So our next speaker is Dr. Aubrey Weigel. Aubrey is a project scientist at Janelia Research Campus who's leading the cell organelle segmentation and microscopy project team. So Aubrey's work really is basically essential to make sense of whatever EM data that you've generated, especially the immense volumes of the FIB-SEM data that you've just seen Sean present. So Aubrey basically uses machine learning and computer vision techniques that she's going to tell us a lot more about today to automatically identify and quantify all of the intracellular substructures that we see in the 3D isotropic EM datasets. Aubrey, please take it away.
All right, and can you just confirm that you can see and hear everything fine?
I can see the screen and I can hear you well. Thank you.
Awesome. All right, well, thank you all for the lovely introduction and for this organization. I'm really excited to share our recent work with you all, automatically segmenting organelles in whole cell volumetric electron microscopy. So a defining characteristic of all living things is that they're made up of cells. Cells are, I think we could all argue, a fundamental unit of life. And focusing on the cell permits us this detailed understanding of all the tissues and organisms that the cell composes. Now, inside of a cell are the organelles, the building blocks. And from highly specialized cells within tissue to plant cells to immortalized cancer cells, even simple unicellular organisms such as protozoa, at the foundational level, all the organelles within these cells have the same basic functions. DNA is packed in the nucleus, proteins are synthesized in the ER, and mitochondria generate ATP to fuel the cell. Historically, light microscopy has brought us a long way in understanding organelles and their functions. The beauty of fluorescence microscopy is it grants us protein specificity. So it allows us to see all the dynamics and how they interact with each other with global context. So we can see all the organelles inside the entire cell. However, with light microscopy, we're limited by protein labels and the resolution. So as an alternative, electron microscopy has been a valuable tool that grants global contrast and higher resolution, but the traditional methods usually limit the volume to which we can investigate, and thus we lose context. And as Sean just shared with you, a valuable tool that kind of gives us the best of both worlds is focused ion beam scanning electron microscopy. So here we get near-isotropic resolution, we get global contrast, and thanks to the ingenuity of the Hess labs and Sean's work, we can do this across large volumes. So not only do we gain resolution, but we also retain context. So here's just some examples of, in addition to what Sean has already showed, of these amazing large volume, high resolution data sets that are being produced here at Janelia. Now, these data sets are absolutely rich with information and they're just begging to be extracted. However, because they are so big and so detailed, extracting that information is a mountain of a task in and of itself. So, for instance, if we zoom in here, and take a closer look, this is a roughly 1 cubic micron volume centered about the centrosome in a HeLa cell. So you'll see popping up here in red the Golgi, and soon the ER. There's vesicles scattered all throughout in orange, and microtubules, the nucleus with its nuclear pores. All of it is really quite beautiful, even with this small cubic micron here. And you might even notice from this small volume that there's this real lack of empty space, even within this volume. And that's actually the problem. So the caveat to all the information that FIB-SEM gives us is that it can make extracting that information even harder to answer the questions that we're really after. So, an example of how long that might take. So for an expert annotator to annotate this block, it took about 7 to 10 days to manually segment all of those organelles and classify every voxel. Now, if we extrapolate that to the entire volume of the cell, it would take a single person 50 to 100 years to segment all of these structures. So obviously impossible. And so enter COSEM. Within Janelia are some of the world's leading experts in machine learning and cell biology and large volume, high resolution EM data acquisition. At their intersect lies this unique area of research. And COSEM's expanding that intersect and driving forward our tools and knowledge with respect to identifying subcellular structures in EM data in an automated way. As a result, the products that COSEM provides includes quantitative analytics and data resources, and it allows us to reimagine our canonical textbook image of the inside of a cell. So it provides biological insight and knowledge, and it does this in a way that is open and accessible to the broader scientific community and to the public. COSEM's a highly collaborative team, including members from scientific computing at Janelia and project technical resources, as well as members from the Saalfeld and the Funky labs. Our pipeline begins after the data has been collected, so with the raw EM data, and it's pre-processed using pipelines that have been developed by Stefan Saalfeld and now in collaboration with Stefan Pribus. So from here, skilled annotators carefully classify each and every voxel within a subblock. These segmentations are then fed into machine learning algorithms as training data, and the prediction output from these algorithms are then evaluated, refined, and in some scenarios used as a tool for other pipelines, such as CLEM registration, for instance. Once we have the predicted whole cell segmentations, we can perform quantitative analytics to get answers on subcellular distributions, interactions, sizes, morphologies. All of this information can then be extracted and acquired. And lastly, all of this data and software is made publicly available and for viewing and download at our online data portal, openorganelle.org. So we'll walk through that pipeline in just a little bit more detail using this HeLa cell as an example. So we'll start here with the raw, with the aligned EM data. And from here, Grace and Allison then manually classify every voxel in subblocks with up to the 37 different classes of substructures that we've defined. So here's an example of what those substructures look like. We have included all the distinguishable membrane-bound organelles, from the popular and easier organelles such as mitochondria and the plasma membrane, to the trickier organelles like ER and Golgi, lipid droplets in the endolysosomal system. We also include cytoskeletal elements like microtubules and nuclear components such as chromatin, the nucleolus, and nuclear pores. And we also annotate the ribosomes. So bringing this all together, you can see here 15 such of these subblocks that Grace and Allison have manually annotated, and the class labels there are shown on the left, color-coded for you. And then all 37 classes and the amount of training data that we have for each is on this graph on the right. So I'll highlight here that we use different combinations of classes for different machine learning setups. So we tested whether training all 37 classes together, which would be all the bars in this graph, versus some, which would be just the green bars, versus individual, which are marked with the white dots, affects the prediction results. So you can also see in the bottom graph here the amount of training data acquired from each data set. So even in this cell, which is arguably has the most training data, only a fraction of a % of the whole cell volume is used for annotation for training data. So the training data I just showed to you is then used to train machine learning classifiers. We use a mostly standard setup with a 3D UNet architecture, as you can see here in the middle. And this is trained by repeatedly feeding in blocks of raw data that produces some output for the innermost part of the block, so the orange part there, which is then compared to what it should look like according to the ground truth annotations. And the parameters of the network are adjusted accordingly. This process is repeated hundreds of thousands of times in our case. And specifically, we train it to predict a signed hyperbolic tangent distance transform of the binary label for each organelle. So this was a iteration of what the Saalfeld lab and Larissa had already done to automatically detect synapses within the fly connectome dataset that Sean had showed earlier. So these distance transforms can be transformed back to a binary segmentation just by thresholding at 0. And here we illustrate this for the mitochondria in blue and the ER in green. But as I mentioned before, we trained a number of these networks where we change how many organelles are actually trained simultaneously. So we have a setup where we train all 37 classes together, one where we include only the classes that are covered by a lot of the training blocks, and then also the setups that just have a few related classes at a time. To find the optimal setup, as well as the iteration for each label and the dataset the networks need to evaluate independent of the training data, we experimented with two different ways of doing this. So the classic approach to this would be to compute metrics on an additional annotation that we, so a holdout block, so acquiring a validation block that you could compare and make metrics against. This is very costly and it comes with its own problems. And because of this, we went with a manual approach, and this would allow us to, one, go more quickly through our evaluations, and two, it would also allow us to go in blindly into new data sets, so it would be more generalizable. This is what this manual evaluation looks like. So to perform these, David implemented a custom Fiji plugin for us to pairwise compare two different predictions at a time. So first, the user selects the two predictions to compare, as well as the organelle class that they want to evaluate. So I'm showing the ER here. And as the movie is showing, a random crop is then chosen from the volume, thresholded at a distance of 0, and then overlaid on top of the raw data. So the user inspects the stacks and selects which prediction, if any, did a better job of predicting the chosen organelle. So note here that the crops are randomly labeled, so the user is blind to which image came from which prediction. The plugin calculates and reports a p-value corresponding to the null hypothesis that the two networks perform equally well. And this evaluation process then repeats for a new crop. Evaluations are performed until after a minimum of 100 evaluations, we can reject the null hypothesis, and the network that performed best is deemed the winner. So with this, no ties are allowed, and that forces us to choose which network performed better. Using the setups optimized via this manual validation, we can then generate predictions on the entire data set, threshold them, and thereby produce the segmentations. So the exciting part. These segmentations aren't perfect yet, though. There's always more work to do, and there's some types of errors that the network makes that are really relatively straightforward to address. So for this, we applied techniques such as size filtering or picking only the largest connected component. So for things like the nucleus, the plasma membrane, and the ER, we could perform these sorts of tricks. Or here on the right, we could improve the instance segmentation with a watershed algorithm. So relatively simple stuff. In the case of microtubules, Nils from Jan Fonke's lab was able to improve the predictions quite a bit by applying a technique he developed that incorporates the structural biases for microtubules. So in this manner, we built up the segmentations all together, and we were able to perform this for 4 different data sets that were our main focus. And what was really exciting about these data sets is that we now had predictions, with these predictions, is that we now had a way to start to see the organelles in a way that we've really never been able to do before. So to exemplify this, we'll take a look at one organelle in particular, which happens to be one of my favorites, the Golgi. So if you look at the textbook shape of a Golgi, it's this cute little stack, and it has 5 to 7 neat little cisternae and a clear cisternal and trans space to it. However, when we look at the segmentations here, what we see is that the entirety of the Golgi in a whole mammalian cell, like we have here, is really more complex. And I would argue it's much more beautiful than what we see in the textbook. So those 7 cisternae that typically you see in the textbook image, in actuality, weave into and ribbon throughout the entire stack. And what may be the first cisternae of one stack becomes the last cisternae of another stack. So you can also see the vesicles studded all around the edges. And furthermore, as I'm showing here, the ER is so intimately connected with the system that it can be really hard to distinguish one from the other. All of these complexities are lost in textbook cartoons and nearly impossible to resolve from light data alone. So it's really exciting to be able to see this level of detail in this large of a volume at once. These segmentations, however, are more than just a pretty picture. We can also start to quantify information from them as well. So for instance, the % volume occupied by each organelle class across different cell types. So I'm showing here, you can see that the nucleus occupies the most space in these cells in magenta, but this is especially so in gercats, which are an immortalized T cell of sorts. Also, you can see the ER occupies a much larger volume in the macrophage cell, which agrees with it being a highly secretory cell. So now I'll walk you through 3 additional analysis examples. So first is the contact site between microtubules and the other organelles in the cell. You can see the microtubule here, and color-coded are the organelles the single microtubule is touching. So this particular microtubule is only about a micron in length, and it's really quite busy. We can quantify these contacts for all the microtubules in the cell, and the results are there in the bottom graph. Most of the contacts occur with the ER, maybe not so surprisingly. Next, I'll show an analysis of membrane curvature. So for this, David Ackerman and the group took ER predictions and iteratively thinned them to produce a medial surface. He then could calculate the curvature on that surface and threshold between planar and green or tubular and purple regions of the ER. Anecdotally, we noticed two things here. So one, a highly secretory cell, such as the macrophage, seemed to have more planar peripheral ER regions than a HeLa cell, and that's quantified in the graph on the left. We also noticed that there seems to be an affinity between planar regions of the ER and the mitochondria contact sites in HeLa cells. So as you can see when the movie pauses, the contact sites in blue seem to match up with the mitochondria in orange. And in fact, when we analyze this for the whole cell, we see that in HeLa cells, the planar regions of ER are most always supported by mitochondria, while in the macrophages, which has a more canonical stacked helicoidal sheets, the mitochondria support is not as readily observed. And let's take this pause, jump forward, hopefully. All right, and for the third analysis example, I'd like to share with you the ribosome classifications that we did. So starting with all the ribosomes, we then classified them as either being ER-bound or cytosolic. And then we further classified them using the ER curvature that they were bound to as to whether they are bound to planar regions of ER or tubular regions. And what we noticed anecdotally is that the ribosomes that were on more planar regions seemed to form polyribosome chains. All right, so everything that I've shown so far was on very high resolution data. So the 4 nanometer isotropic data that Sean spoke about at the end of his talk. However, we didn't want to restrict ourselves to this high resolution data because most of the FIB-SEM data is still acquired at 8 nanometers. So what we were able to do is reuse our 4 nanometer training data to train a second set of networks where we roughly simulate what 8 nanometer versions of that raw data would look like by randomly subsampling the data on the input side. So with those networks, we were able to produce 4 nanometer segmentations on 8 nanometer data, so sort of super resolution predictions, if you will. So on the bottom here is another HeLa cell imaged at 8 nanometers, which should be pretty similar to our training data. And the segmentation seems to work there, so that's nice. But what's really encouraging is that we're able to get decent results on this mouse choroid plexus data set on the top, which is chemically fixed tissue, so quite different from the samples. It's chemically fixed and it's tissue, so it's quite different from all the samples that we've trained on, which is really encouraging. Another interesting aspect is that the 8 nanometer networks allowed us to get segmentations for existing correlative light and electron microscopy datasets, such as published by Hoffman et al. So here, John was able to use them to automate the alignment of the EM data to the light microscopy data, such as in this COS7 cell. So as we're zooming in here, what you'll see is the deformable part of that alignment as we're switching between the original FIB-SEM and the FIB-SEM that's aligned to the light data. So what John specifically did here was he used the mitomembrane segmentation, which we produced, which will show up in orange, to generate images that look like the light microscopy data in blue. And he did this by downsampling and applying an anisotropic Gaussian blur to mimic the PSF of the light microscope. So those images are then automatically aligned to the light mitos. And as you can see, they match up quite nicely. And then as a byproduct, the ER channels are aligned. So the ER segmentation shown in green and the light data shown in, I think it was in blue or purple, sorry. So I should mention here that all of these is sort of the best case scenario. So the extent to which the alignment process can be automated really depends on the quality of the data. And sometimes we'll have to manually tweak things, right? If a cell is untransfected and it doesn't show up in the fluorescent channel, but it shows up in the EM channel, so those will have to be removed. But I can say having to had aligned several of these data sets with and without predictions, even in the worst case scenario where you have to do things manually, having the predictions to align to really does speed the process up from taking days to just a couple hours, which is quite encouraging. So most importantly, all of this work is being made publicly available through our open access portal, Open Organelle, with the data hosted by AWS. Here, together with the HES Lab and FIB-SEM technology group, we provide a browsable and soon-to-be queryable interface for people to explore these EM datasets and the accompanying segmentations. So you can browse pre-made views, or you can customize your views based on what you want to see. You can then explore the data online using NeuroGlancer. The data is set up in a multi-resolution pyramid scheme, which allows for fast online browsing. And we also provide tools for anyone to download any or all of the data if they so dare, because these are rather large data sets. And that includes the raw data, the training data, the segmentations, and the analysis. So the Salfeld Lab has also implemented in 5 capabilities in Fiji. So this means that anyone can open Fiji and read directly from the Amazon servers these data sets. So there is no need to download these if you don't want to. We also provide tutorials, and we've provided a glossary of sorts containing a definition and an example of each object class. And I'm showing here the centrosome for a Jurkat cell, I believe. So, and then we link out to our GitHub repositories where all of these tools and models and source code is available as well. So of course, the ultimate goal of COSM is to segment organelles and EM data. And there's still a lot of work to be done here. This is only just the beginning, which is really exciting. But what we now have in place is a pipeline to get us started. And while, of course, we're trying to continuously improve every aspect of this, we're also seeking to understand what else is needed to maximize the impact of this work. So for instance, how much training data is needed for a new data set, or how much proofreading is required, how generalizable are analysis methods in new data sets. So we're branching out more to help answer some of these important questions. We're collaborating with the AIC, who has their own FIB-SEM and CryoClem system now, transferring tools and knowledge. We're continuing to generate and integrate more training data from a variety of data sets, a majority of which includes tissue. And we're also applying our analysis methods to help quantitate segmentations from outside groups. Because let's be honest, even if you have the segmentations in hand, that really is only the beginning of the work that needs to be done. Finally, and importantly, all this work was really made possible by an incredible and collaborative team effort. And you can find this in our preprint now on bioRxiv. And really, it would not be possible without the larger support of administration, labs, and support teams that are available here at Janelia. And with that, I thank you for your time, and I'm open for questions.
Thank you so much, Aubrey. That was truly wonderful. I'm going to start with 2 comments that I think most of us resonate with. These comments come from Christoph and Michael. This is so cool. That's the first comment. And the second one from Michael is, this is the image analysis dream, which I think I concur with. In terms of the questions, Nico is asking, are you using the same neural network to predict all the cell types that you've shown here, or they're independently trained and then predicted?
Yes. So, so I mentioned kind of briefly, sorry, there's a lot to go through in this time, but we do have different neural networks for training all the classes together, a subset, or individual. And so those were really the main parsing that we did, but all of the training data got pooled together. We are doing separate experiments to see how much one cell influences that training, but we don't quite have those results yet. But to date, everything has been pooled together.
So you mentioned using or training on a high pressure frozen sample and then testing it on a chemically fixed sample. Obviously, the signal to noise ratios are drastically different and the morphologies may be altered. How robust are the predictions? I know you said it works, but would you actually use it, or would you want to retrain it?
So I think we're at the stage right now where it's a per data set experiment, right? And really, it has less to do about what the cell type is and more to do about what the data looks like. So the signal to noise level, like you mentioned, the amount of staining for, and not even really the staining for each organelle, because what was surprising to us is when the whole cytoplasm is cleared, like in a chemically fixed cell, that really didn't seem to mess up predictions that much. But what we'll notice even from one high pressure frozen cell to another is that if suddenly a mitochondria has very dense cristae to the point where you can't discern them, then the networks will have a harder time picking out those organelles. But I think we're at a point right now where the solution to a lot of our problems is just more training data. Maybe, so actually, unfortunately.
That's a good segue to Thomas's question, is asking about ribosomes, specifically in the context of how easy is it to segment mitochondria-bound or associated ribosomes?
Super easy, right? So we have the mitochondria, we have the ribosomes, and really it just becomes a contact site problem. So it would just be a matter of running that analysis. Yeah.
And then George is asking, how can this data be accessed and along with the visualizations?
Yeah, so everything's on Open Organelle. So if you go there, you can go through the data sets. There's instructions there on how to open them in Fiji then if you want to. Everything you can look at through NeuroGlancer. And the website is new and young, so please be kind, but also provide constructive feedback because we're always looking to improve things.
And the last question comes from William. He's asking if someone were to have a 4 nanometer or 8 nanometer FIB-SEM data and is annotating keratin and other structures that are not part of the network that you've already trained, would COSEM be interested in those annotations, presumably?
Yes. Yeah, I would suggest reach out to me directly, or go to Open Organelle, and there is an email address there under the About page, cosemdata@janelia.org, where you can get in contact with us and we can discuss further.
Thank you so much, Aubrey. Again, on behalf of all of the participants, I'm going to be the one applauding your mind-blowing talk. Thank you again.
Thank you.